Hello everyone, let's have a quick recap about carbohydrates, their categories, what are actually carbohydrates, all those related information. Let's start with carbohydrates. Carbohydrates are the most abundant organic molecules in nature. They are primarily composed of elements carbon, nitrogen, and oxygen. The name carbohydrates literally means hydrates of carbon. Some of the carbohydrates possess the empirical formula CH2O raised to N, where N is less than or equal to 3. Satisfying that these carbohydrates are in fact carbohydrates. However, there are several non-carbohydrate compounds, acetic acid, C2H4O2, lactic acid, which also appear as hydrates of carbon. Further, some of the genuine carbohydrates, rhamnohexose, deoxyribose, do not satisfy the general formula. Hence, carbohydrates cannot be always considered as hydrates of carbon. Carbohydrates are formed in the plants by photosynthesis from carbon dioxide and water in the absence of sunlight. NCO2 plus NH2O plus energy gives CnH2n plus CnH2nOn plus NH2O. So carbohydrates may be defined as polyhydroxyaldehydes. Polyhydroxyaldehydes or ketones. Polyhydroxy ketones or polyhydroxyaldehydes or compounds which produce them on hydrolysis. The term sugar is applied to carbohydrates soluble in water and sweet to taste. Classification of carbohydrates. Let's learn about the classification. Carbohydrates are often referred to as saccharides. In the sense, the Greek word saccharon means sugar. They are broadly classified into three major groups: monosaccharides, oligosaccharides, and polysaccharides. This categorization is based on the number of sugar units. Mono and oligosaccharides are sweet to taste, crystalline in character, and soluble in water. Hence, they are commonly known as sugars. What does the word oligo mean? Oligo means few. So, let's learn about monosaccharides. Monosaccharides, mono in the sense one, are the simplest group of carbohydrates that are often referred to as simple sugars. They have the general formula CnH2n H2ON and they cannot be further hydrolyzed. The monosaccharides are divided into different categories based on the functional group and the number of carbon atoms. Aldoses are there. Aldoses. When the functional group in monosaccharide is an aldehyde, monosaccharides, mono 1, are the simplest group or carbohydrates that are often referred to as simple sugars. They have the general formula which we have already told and they cannot be further hydrolyzed. The monosaccharides are divided into different categories based on the functional groups and number of carbon atoms. First one is the aldoses. When the functional group in monosaccharide is an aldehyde, they are known as aldoses. Example, glyceraldehyde and glucose. Then the ketoses are there. When the functional group is a keto group, they are referred to as ketoses. For example, fructose is a ketose and deoxyacetone is also a ketone. Based on the number of carbon atoms, the monosaccharides are regarded as triose, tetroses, pentoses, hexoses, and heptoses. These terms along with functional groups are used while naming monosaccharides. For instance, glucose is an aldohexose while fructose is a ketohexose. Monosaccharides, when we just look at the empirical formula of the monosaccharides, we can see trioses are there, tetroses, pentoses, hexoses, and heptoses will be there. And there are some aldoses like glyceraldehyde, erythrose, ribose, xylose, arabinose, glucose, galactose, mannose, glucoheptose, and some ketones will be there like dehydroxyacetone, erythrulose, ribulose, cellulose, fructose, pseudoheptulose, etc. So when we just represent aldoses and ketoses, we just first write a carbon, then put a hydrogen there, and then put a double bond into the top, and next carbon will be next to that, and put OH on the right side and H on the left side, and on the bottom we will be putting CH2OH. In this case, the central one, the carbon with two double bonded O, is a C1, that is the anomeric carbon. That one is the anomeric carbon. So there will be a aldehyde group. So that's why this particular structure is called as an aldose. When we are trying to draw a ketose, first we will be putting a carbon, then another carbon in the top of that, and then they are linked to each other by a bond. Then the top carbon has an CH2OH. The first carbon has a double bond and O. Then we will be drawing another carbon in the bottom with an OH on the right side and a H on the left side, along with the CH2OH on the bottom. Okay, so in this case we can see a C double bond O. That is a keto group attached to C2 anomeric carbon. Keto group. So this is the structure of a ketose. Okay, that is the difference between an aldose and a ketose. And there are many different structures like diglyceraldehyde, erythrose, triose, ribose, arabinose, xylose. All those are very important. And ring structure of monosaccharides, when we are going to learn, let's explain all those terms associated with the ring structure of monosaccharides. In aqueous solution, monosaccharides containing 5 or more carbon atoms exist mostly in ringed structures, either as 5-membered ring in case of furanose ring or 6-membered ring like a pyranose ring. Alcohols react readily with aldehydes to form hemiacetals. Write down hemiacetals. The linear form of glucose and other aldohexoses can undergo a similar intramolecular reaction to form a cyclic hemiacetal. The resulting 6-membered oxygen-containing ring is similar to pyran and is designated as a pyranose. Pyranose and a furanose. For example, the 6-membered ring of glucose is called glucopyranose. Okay. In a similar manner, ketones can react with alcohols to form hemiketals. The analogous intramolecular reaction of a ketose molecule such as fructose yields a cyclic hemiacetal. The 5-membered ring has formed is reminiscent of furan and is referred to as a furanose. Earlier we were talking about pyranose. Here we are going to learn about furanose. For example, a 5-membered ring of fructose is called fructofuranose. The cyclic pyranose and furanose forms are the preferred structures for monosaccharides in aqueous solution. At equilibrium, the linear aldehyde and ketone structure is only a minor compound of the mixture, generally much less than 1.2%. So first we can easily recall the first name, that is the glucose. Glucose is a simple sugar with the molecular formula C6H12O6. Glucose is the most abundant monosaccharide. Glucose is mainly made by plants and most algae during photosynthesis from water and carbon dioxide using energy from sunlight, where it is used to make cellulose in cell walls. That is a new information. Glucose is used to make cellulose in cell walls, which is the most abundant carbohydrate. In energy metabolism, glucose is the most important source of energy in all organisms. Glucose for metabolism is stored as a polymer in plants mainly as starch and amylopectin, and animals as glycogen. In case of animals, glucose is stored as glycogen. Glucose circulates in the blood of animals as blood sugar. The naturally occurring form of glucose is D-glucose. Glucose is a monosaccharide containing 6 carbon atoms and an aldehyde group, and is therefore an aldohexose. The glucose molecules can exist in an open chain, acyclic as well as ring cyclic form. D-glucose is the naturally occurring form. So glucose is a monosaccharide containing 6 carbon atoms. So it will be a aldohexose. In the sense there will be an aldose and a hexose. So aldohexose. there will be an aldehyde group along with a hexose ring. So it's a aldohexose. The glucose molecule can exist in an open chain acyclic as well as ring cyclic form. Glucose is naturally occurring and is found in fruits and other parts of plants in its life state. In animals, glucose is released from the breakdown of glycogen in a process known as glycogenolysis. Glycogenolysis. By the process of glycogenolysis, glycogen is breakdown and glucose is released from the liver. That process in animals is called glycogenolysis. There are two enantiomers of glucose. When we draw a glucose molecule, when we are drawing D-glucose, as in the sense D-glucose is the most abundant form. So first we will be putting a chain of 6 carbons one by one. 1, 2, 3, 4, 5 and 6. Then we will be drawing a double bond in the first carbon and putting an O. On the other side an H. Then the second carbon we will be putting an OH on the right side and H on the other side. But next carbon we are going into the downside, so then we'll be putting a H in the right side and OH in the left side. And then we will be putting OH to OH on the right sides and then H on the left side. And in the bottom we will be putting a CH2OH. That is the structure of a D-glucose. In aqueous solution, glucose exists mostly as ring structures, either 5-membered ring, furanose ring or 6-membered pyranose ring. The formation of this ring structures is the result of a general reaction between alcohols and aldehydes to form derivatives called hemiacetals. Thus, the aldehyde group of glucose at C1 reacts with the alcohol group at C5 to form two types of cyclic hemiacetals, namely alpha and beta anomers, alpha D-glucopyranose and beta D-glucopyranose. Alpha D-glucopyranose and beta D-glucopyranose will be there. When we learn about fructose or fruit sugar is a simple ketonic monosaccharide found in many plants, where it is often bound to glucose to form a disaccharide sucrose. So when we combine glucose along with fructose, that is the composition of sucrose. Okay, sucrose contains glucose and fructose. It is one of the 3 dietary monosaccharides along with glucose and galactose that are absorbed directly into blood during digestion. Pure dry fructose is a sweet, white, odorless, crystalline solid and is the most water-soluble of all the sugars. Fructose is found in honey, tree and vine fruits, flowers, berries and most root vegetables. Commercially, fructose is derived from sugar cane, sugar beets and maize. High fructose corn syrup is a mixture of glucose and fructose as monosaccharides. All forms of fructose, including fruits and juices, are commonly added to foods and drinks for palatability and taste enhancement and for browing of some foods such as baked goods. Excessive consumption of fructose, especially from sugar-sweetened beverages, may contribute to insulin resistance. Insulin resistance is a probable result. Obesity, elevated LDL cholesterol and triglycerides leading to metabolic syndrome. When we consume fructose excessively, some difficulties may arise like the insulin resistance, obesity, elevated LDL cholesterol and triglycerides. All this lead to metabolic syndrome. Fructose is a monosaccharide containing 6 carbon atoms and a keto group and is therefore a ketohexose. The fructose molecule can exist in the open chain as well as ring form. Two enantiomers of fructose can be seen, D-fructose and L-fructose. In aqueous solution, fructose exists mostly as ring structures, either 5-membered ring, furanose ring, or 6-membered ring, pyranose ring. So furanose ring will be there and a pyranose ring will be there. Furanose ring and pyranose ring. The formation of these ring structures is a result of a general reaction between alcohols and ketones to form a derivative called hemiketals. Thus, the ketal group of fructose at T2 reacts with alcohol groups at C5 to form two types of cyclic hemiketals, namely alpha and beta anomers, alpha D-fructofuranose and beta D-fructofuranose. Next, let's learn about galactose. Galactose is a monosaccharide sugar that is about as sweet as glucose and about 65% as sweet as sucrose. It is an aldohexose and a C4 epimer of glucose. Galactose is a C4 epimer of glucose. Okay. A galactose molecule linked with the glucose molecule forms a lactose molecule. So, a galactose plus glucose gives a lactose. Okay. The hydrolysis of lactose to glucose and galactose is catalyzed by the enzyme lactase in humans and mammals and beta-galactosidase in bacteria. Galactone is a polymeric form of galactose found in hemicellulose and forming the core of the galactones, a class of natural polymeric carbohydrates. Galactones exist in both open chain and cyclic form. 4 isomers are cyclic, 2 of them with a pyranose 6-membered ring, 2 with a furanose 5-membered ring. Galactofuranose occurs in bacteria, fungi, and protozoa. In the cyclic form, there are 2 anomers, namely alpha and beta. In the beta form, the alcohol group is in the equatorial position, whereas in alpha form, the alcohol group is in the axial position. So what's happening here? Galactofuranose occurs in bacteria, fungi, and protozoa. In the cyclic form, there are 2 anomers, namely alpha and beta. In the beta form, the alcohol group is in the equatorial position. Don't forget, in beta form, it is in the equatorial. Alcohol group is in the equatorial position. Alcohol group is in the equatorial position. Whereas in the alpha form, the alcohol group is in the axial position. Next major one is the mannose. Mannose is a monosaccharide of the aldohexose series of carbohydrates. It is a C2 epimer of glucose. Earlier we have learned about galactose, which is a C4 epimer of glucose. This is mannose. Mannose is a monosaccharide and C2 epimer of glucose. Mannose is important in human metabolism, especially in the glycosylation of certain proteins. N-linked glycosylation, which is a post-translational modification of proteins. N-linked glycosylation, which is a post-translational modification of protein. Several congenital disorders of glycosylation are associated with mutations and enzymes involved in mannose metabolism. Mannose is not an essential nutrient. It can be produced in the human body from glucose or converted into glucose. Mannose provides 2 to 5 kilocalorie per gram. It is partially excreted in the urine. Mannose commonly exists as 2 different sized rings, the pyranose 6-membered form and the furanose 5-membered form. Each ring closure can have either an alpha or beta configuration at the anomeric position. The chemical rapidly undergoes isomerization among these 4 forms. D manopyranose will be there. Alpha D manopyranose and beta D manopyranose, and alpha D manofuranose, beta D manofuranose. This four undergo chemical reactions, isomerization reactions. Next is the ribose. Ribose is a pentose. Ribose is a pentose sugar with molecular formula C5H10 and O5. In the naturally occurring form, D-ribose is a component of the ribonucleotides from which RNA is built, and so this compound is necessary for coding, decoding, regulation, and expression of genes. Coding, decoding, regulation, and expression of genes. Okay. It has a structural analogy, deoxyribose, which is a similarly essential component of DNA. Like most sugars, ribose exists as a mixture of cyclic forms in equilibrium with its linear form. D-ribose and L-ribose will be there. Like most sugars, ribose exists as a mixture of cyclic forms in equilibrium with its linear form, and is readily interconvert, especially in aqueous solution. Like most sugars, ribose exists as a mixture of cyclic forms in equilibrium with its linear form, and is readily interconvert, especially in aqueous solution. In its linear form, ribose can be recognized as the pentose sugar with all of its hydroxyl functional groups on the same side of its Fischer projection. D-ribose has these hydroxyl groups on the right hand side, and while L-ribose has its hydroxyl groups appear on the left side on the Fischer projection. Cyclization of ribose occurs by hemiacetal formation due to attack on the aldehyde of the C4 hydroxyl group to produce a furanose form, or by the C5 hydroxyl group to produce a pyranose form. In each case, these two possible geometric outcomes, named as alpha and beta, are known as anomers. Depending on the stereochemistry, at the hemiacetal carbon atom, the anomeric carbon, at room temperature, about 76% of D-ribose is present in pyranose forms and 24% of the furanose forms, with only about 0.1% of the linear form present. Alpha D ribopyranose, beta D ribopyranose, alpha D ribofuranose, and beta D ribofuranose will be there. So that's all about carbohydrates, especially monosaccharides, and we can learn about oligosaccharides and different types of oligosaccharides, all the stuff later.